All right, in this video, we're going to talk about the process, the stages of meiosis. You'll see some similarities between mitosis and meiosis. So, first of all, meiosis is divided into two sets of divisions, meiosis 1 and meiosis 2. And so your cell is going to be in interphase when we start. It's going to go through prophase 1. We're going to have 2 copies of each chromosome for a total of 4. Metaphase 1, they line up. Anaphase 1, the chromosomes separate, very much like mitosis. Then telophase 1, the cell splits, and then you end up with, again, we now have 2 chromosomes. So we're doing a reduction division here, where we're going through the different stages of meiosis 2, prophase, metaphase, anaphase, telophase. At the end, you end up with 4 cells, which are haploid, have 1 copy of each chromosome. So the difference is we don't have a doubling of the chromosomes between meiosis 1 and meiosis 2. So like I said, 2 divisions. So you have DNA replication in interphase of meiosis 1. First division of meiosis separates homologous pairs of chromosomes. Second division separates sister chromatids. So to prepare for meiosis, just like for mitosis, you've got to duplicate the DNA. Good. So why do we do this? Meiosis has evolved from mitosis, so that step is kind of conserved from mitosis. We kind of use the same machinery for meiosis that the cell uses for mitosis. So again, you have a DNA replication, just like with mitosis. So first division, like I said, is separation of the homologous chromosomes. Go from a diploid cell, basically, to a cell that has 4 copies of each chromosome. Tetrads are what we call those. So one of the things that happens in meiosis 1 is something that helps rearrange the DNA. We'll talk more about that in a later video. But synapsis or crossing over occurs. Again, you have 4 single-stranded chromosomes at the start, and you have 4 double-stranded chromosomes as we go into meiosis and end up with 4 single-stranded chromosomes at the end. So between meiosis 1 and meiosis 2, there's no copying and no duplication of the DNA. This is where the sister chromatids are going to separate. We got one double-stranded chromosome at the end here. Then we have the end of meiosis, you end up with single-stranded, 1 copy each chromosome. This is very much like mitosis. But instead of mitosis, we end up with 4 daughter cells, and they're not genetically identical because of some rearranging that goes on. And it's only going to be sperm or egg cells produced this way. Here's kind of just a review of what this looks like here. Okay. Here's some actual photographs of the